All right, chapter 6, we covered genetic analysis in bacteria and phages, and this will be the last chapter on the first exam, on the material covered for the first exam. So this chapter is going to be a bit of a departure. We're going to move on from eukaryotic organisms to the strange world of prokaryotes and viruses. So here are the goals, the key concepts, to explain how bacteria phages are grown and phenotyped in the lab. So this is very different from what you're used to. What is a phenotype of a bacteria? What is a phenotype of a phage? And then also how you can grow these things in mass amounts in the lab. Identify and understand the different ways that genetic recombination can occur in bacteria and phages. So sex, as we think about it, does not happen in bacteria. Two bacteria typically aren't required to have sex in order to have progeny, but genetic recombination can occur. And this can either be through processes controlled by the bacteria or through just random processes that occur using the phages. And then finally, interpret the results of gene transfer experiments in bacteria and phages. So, what is a bacteria? So, bacteria are pretty much the most successful kingdom of life, just in terms of the raw number of species. There are all sorts of species of bacteria, and they pretty much live everywhere. So you can find them within your body, you can find them within the soil, you can find them within streams, you can find them in the air. Pretty much bacteria have been able to colonize every single environment known to man. Typically, bacteria are haploid, and they have a singular circular chromosome. So that's one of the important things to remember about bacteria, is that they're not diploid, so they only have one copy of each gene. They also do not have linear chromosomes, so they do not, they do not condense their chromosomes in the same way that eukaryotic cells do. Rather, the DNA is distributed throughout the cell body, even during replication and when it's segregated to different cells. Bacteria can also possess plasmids. So plasmids are outside of their normal chromosome, and these are very small circular DNA molecules. So plasmids, there can either be a single plasmid per cell, there can be a handful of plasmids per cell, or there can even be hundreds to thousands of plasmids per cell. So it's very varied depending on the exact plasmid and kind of the ways, the molecular mechanisms of replication that plasmid uses. So plasmids are small circular DNA molecules. They can also contain genes. They also typically encode a small number of genes. And these plasmids are useful for things like antibiotic resistance, is one of the reasons that we know so much about them. And they're also very important for genetic experiments in the lab. So kind of if you want to do genetic manipulation, especially 50 years ago, starting 50 years ago, plasmids was the way to do that. And still today, plasmids are pretty much the go-to way for any sort of genetic manipulation of bacteria. Bacteria reproduce asexually, so that means that they don't have to find a partner in order to reproduce. And so a single bacteria cell is able to divide into a mostly identical daughter cell. So genetically, by identical, this means genetically identical. So each cell will have an approximately the exact same DNA, because each cell will have that singular circular chromosome. The only thing that would be different would be any sort of replication errors, DNA replication errors that occur. So unlike humans, humans are diploid, and one of our goals is to have genetically distinct progeny, and so we accomplish that using meiosis. That is not a goal of bacteria. So bacteria reproduce pretty much so that each cell is identical to each other at the DNA level. So plasmids might be a little bit different. They might inherit a different amount of plasmids. One cell might have 12 plasmids and another cell might have 8 plasmids. Or if it's a very low copy, one cell might have 3 plasmids and the other cell might have 0 plasmids. Just because they are identical to each other does not mean that the daughter cells look identical to each other. So a very famous example of this is what I actually studied in my graduate school. This is a bacteria called Caulibacter crescentus. and it's called that because of, one, its stalked shape. So the calo, like cauliflower, it's a stalked bacteria. And then the crescentus comes from the fact that it's not a line, rather it seems to form a crescent. But one thing you'll notice about that bacteria is that the daughter cell is distinct from the mother cell. So the mother cell was the stalked cell, but the daughter cell, instead of having a stalk, actually has a flagellum, which is used for swimming. So this bacteria is able to swim away. So this bacteria is, rigidly affixed to something. So it's usually found in streams. So maybe it's attached to a pebble, and this bacteria is free to swim away to kind of find a new environment using the flagellum. So even though they're genetically identical, that doesn't mean that phenotypically they have to be exactly the same. And bacteria actually come in all sorts of shapes. So this is an example of a cubic bacteria found in the Dead Sea, where salt concentrations are very high. So it's thought that this shape is an adaptation to this high salt content. And you can also find them, for example, in soil, in your gut, etc. So bacteria also can be infected by viruses, just like us. Bacteria have viruses that target them, and these viruses are referred to as bacteriophages. So we're going to refer to phages throughout this chapter. So when I talk about phages, it's kind of it's the same thing as calling it a virus. And then one final thing that distinguishes bacteria is that they don't have a nucleus. So their DNA is within the same cytoplasm as kind of the rest of the ribosomes, as the rest of the proteins, etc. So this chapter, there's going to be 4 parts of it. So one, we're going to discuss growing and phenotyping bacteria and phages. How do we do that? Part 2 and part 3 will be on genetic exchange between bacteria. So how are they able to exchange different types of DNA with each other? And there's 3 main processes. So the first 2 will be on conjugation and transformation, and then the part 3 will be on transduction. And then finally, part 4, we'll talk a little bit about genetic exchange in viruses.